Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Frizul Faiz bin Abdul Aziz. F20A0459 will present about serological test. Firstly, the scientific study of blood serum and other body fluid is known as serology. Serology is the diagnostic detection of antibodies in the serum according to immunological practice. Blood serum and bodily fluid like semen and saliva are used in serological tests. In actual usage, the phrase typically relates to the diagnostic identification of serum antibodies or the detection of infectious agent antigens. Such antibodies are frequently produced in response to an infection against a specific germ, to the body's own proteins in case of autoimmune disease, or to other foreign proteins, for example, in response to an unmatched blood transfusion. Next, a B cell's initial exposure to an antigen triggers a basic immunological response. The generation of antibodies that can bind directly to the antigen is stimulated when the antigen binds to the surface of the B cell. There is a temporary lag in the body's ability to combat the invasive antigens, since this first identification phase requires time for antibody production. The main immune response produces an antibody called immunoglobulin M, that is IgM, which is important in battling infection. Large amounts of IgM are created when an antigen is first introduced into the body. IgG, a highly specific immunoglobulin, is being produced by B cell more slowly in the meantime. Hence, the capacity of IgG to bind to antigen molecules more tightly makes it play a greater role in the elimination of antigens from the body after IgG is created in large quantities. The amount of circulating IgM increases quickly in the bloodstream during an infection. IgM levels then start to drop as IgG concentrations rise. Measuring the ratio of IgM to IgG in the circulation allows medical lab staff to determine the kind and length of an infection. When the ratio of IgM to IgG is high, the infection is in its early stage. When the ratio of IgM to IgG is high, the infection is in its later stage. As you can see, the immune response when B and T cells start to divide, some of their progeny will develop into memory cells with a lengthy lifespan. This memory cell will retain details of every unique infection the animal came into contact with over its existence, enabling it to mount a potent defense should it ever reinvade the body. That's all from me.
The one for today is my part will be showing the types of serological tests. First is enzyme-linked immunosorbent assay, known as ELISA. ELISA is widely used serological test. It detects and measures specific antibodies or antigens in the blood. The test involves the binding the antibodies in the sample to a solid surface, adding an enzyme linked detection antibody to produce a measurable signal. ELISA is valuable in diagnosing infectious disease and monitoring immune response. Second is rapid diagnosis tests. Rapid diagnosis tests are point-of-care serological tests designed for quick result. They provide rapid detection of specific antibodies against pathogens like HIV, malaria, or COVID-19. Rapid diagnosis tests often utilize lateral flow technology similar to pregnancy tests to detect the presence of antibodies. These tests are convenient for field testing and emergency situations where immediate results are crucial. Next is immunofluorescence assays, IFA. IFA employ fluorescence dye to detect and visualize specific antibodies. It labels the antibodies with fluorescent molecules and examine the sample under a fluorescent microscope. The presence of fluorescence indicates the presence of antibodies. IFA is valuable for diagnosing autoimmune disease and detecting certain viral infection. Next is neutralization assay. Neutralization assays measure the ability of antibodies to neutralize pathogens. They assess the effectiveness of vaccine and evaluate the immunity to specific viruses. By determining the neutralizing capacity of antibodies, this provides insight into immune protection. Neutralization assays are vital tool in vaccine development and monitoring. Western blot. Western blot is a conformity test antibody detection. It involves separating a protein from a blood sample using gel electrophoresis, testing them onto a membrane separated protein or probe with specific antibodies to detect target antibodies. Western blot is commonly used to confirm the presence of HIV antibodies. So in conclusion, serological testing plays a significant role in diagnosing infection, assessing immune response, and monitoring vaccine effectiveness. We have covered the key types of serological testing, including ELISA, RPTs, AFA, neutralization assays, and Western blot. Choosing the appropriate test depends on the specific disease and purpose of testing. Understanding this testing method enhances our ability to combat infectious disease and protect public health. That's all for me. Thank you.
Method of serological testing. Serological methods are used for measuring the antibody response, while the presence of virus can be demonstrated by cultivation or demonstration of specific antigen or gene sequence. Several methods of serological testing. First, indirect agglutination. Indirect agglutination helps to determine the type of antigen isolated from the patient using non-serum antibodies. Direct agglutination. Direct agglutination is a method in which antibodies in patient serum are determined by adding a suspension of non-bacteria. Antigens are found on the surface of bacteria. Binding of the antibody to the O antigen, the polysaccharide chain of the liposaccharide, causes a compact precipitate to form. Flagella H antigen form a cloud resembling fluffy cloud or misty glass. Direct agglutination is used to detect Salmonella in typhoid fever, Brucella bacteria, and Rickettsia. Direct hemagglutination. In direct hemagglutination, the surface molecules of the erythrocyte are antigenic molecules. The positive reaction, which manifests itself the agglutination of red blood cells, is caused by the binding of antibodies produced by ferret organisms to this structure. An example is the Paul-Bunel reaction to detect infectious mononucleosis. Next, hemagglutination inhibition or HIT is used in influenza virus and other hemagglutinin-bearing disease. Virus-bound antibody inhibit hemagglutination. Precipitation. Upon precipitation, an insoluble complex is formed by the reaction of the antibody with a soluble colloidal antigen. A suitable medium is an aqueous solution or an agar or agarose gel. There are three types of precipitation. First, precipitation in solution. Second, gel precipitation. And lastly, double arrangement according to Auto-Loney. Next, complement fixation. Complement fixation reactions are based on complement properties. Free complement lysis erythrocyte and is capable of binding to the antigen-antibody complex. Once complement complex, it can no longer disrupt erythrocyte. In a patient blood serum sample, complement must be inactivated by temperature. And lastly, neutralization. In neutralization reaction, the properties of antigen are inhibited. In the case of viral infection, the presence of antibodies is manifested, for example, by their inability to infect the experimental animal. The neutralization reaction therefore include the above mentioned HIT. Another use is in the detection of anti-streptolysin O antibodies against streptolysin O in Streptococcus type A, C, and G.
What happened during and after serological testing? During serological testing, a sample of blood is taken from a person and tested for the presence of specific antibodies. Here is a step-by-step breakdown of what happened during and after serological testing. First is sample collection. A healthcare professional collects a blood sample from the person, either through a finger prick or by drawing blood from a vein. Next is laboratory testing. The blood sample is sent to a laboratory where it undergoes testing. There are different types of serological tests such as enzyme-linked immunosorbent assay or ELISAs or rapid diagnostic test RDT, which detect the presence of antibodies in the blood. Third is test analysis. The sample is analyzed in the laboratory to detect the presence of specific antibody related to particular infection or disease. This can help determine if the person has been previously exposed to the infection or if they have developed immunity. Next is test result. Once the analysis is complete, the test results are generated. The result may indicate whether the person has antibodies against the infection or disease being tested for. Positive result suggests past exposure or immunity, while negative result indicate the absence of antibodies. And last one is interpretation and counseling. A healthcare professional interprets the test result and discuss them with the person. They may provide information and counseling based on the test outcome, which could include guidance on next step, further testing, vaccination, or precaution to be taken. After serological testing, there are several possible outcomes. First one is positive result. A positive result indicates the presence of specific antibodies in the blood, suggesting past exposure to the infection or disease. Depending on the situation, this may mean the person has developed immunity. Next one is negative result. A negative result means that no specific antibodies were found in the blood. This could indicate that the person has not been exposed to the infection or has not developed antibodies yet. It does not necessarily rule out the possibility of recent infection, as antibodies may take time to develop. Next one is further testing. In some cases, further testing may be recommended to confirm the presence of antibodies or to test for other infection or disease. Last one is immunity assessment. Serological testing may be used to evaluate the level of immunity in a population or to determine if someone has sufficient immunity for certain purpose, such as blood donation or return to work. It's important to note that serological testing has limitations and should be interpreted in conjunction with clinical evaluation and other diagnostic methods. Consultation with healthcare professional is crucial for accurate interpretation and appropriate management.
Serological testing is a type of medical testing that involves analysis patient blood serum that detect the presence of antibody against various pathogens such as bacteria or virus. The result of serological testing can provide valuable information about patient immune response to a infected or vaccination and can also be used to diagnose certain disease. Serological testing is provide valuable information about patient immune response to a pathogen. The following are the results that can be obtained. First is positive IgM. A positive IgM result indicates that the patient has been infected with a pathogen and that their immune system is currently produced antibody to fight the infection. IgM antibodies typically appear in a blood with 1 or 2 weeks after the infection and may present to several months. Positive IgG. A positive IgG result indicates that the patient has been infected with a pathogen in the past and that their immune system has produced antibodies to provide long-term protection against future infection. IgG antibodies typically appear in blood several weeks after the infection and may persist for several years. Positive IgA. A positive IgA result indicate that the patient has been infected with a pathogen and that their immune system is currently producing antibody to fight the infection. IgA antibodies are typically found in the mucous membrane of the respiratory and digestive tract and provide localized protection against pathogens. Elevated titers. An elevated titers refer to high levels of antibody that patient blood indicate there a risk of past infection that a pathogen. A rising titer over time may also indicate that active or ongoing infection. Lastly is negative results. A negative results indicate that the patient has not been infected with pathogen. However, it is important to note that some patients may be seronegative despite being infected with a pathogen, especially in the earlier stage of infection. For the conclusion, positive result indicate an active or past infection, while negative result may indicate the absence or infection need for future testing. That's all from me. Thank you.